Hello everyone! Today we're going to show you the cell cycle and meiosis. The first stage of the cell cycle is interphase, which consists of G1, S, and G2. In G1, cells grow, develop, and differentiate. This also includes the G0 checkpoint, which determines whether or not the cell will divide. The next phase is the S or synthesis phase. During this phase, DNA is copied. The last phase is G2, which is when the cell prepares for replication by making proteins, storing food, and replicating organelles. Then we have the G2 checkpoint, which is when the cell checks for DNA damage, and if there is damage, it will try to fix it. If it can, it will undergo apoptosis, or programmed cell death.
After interphase, the cell will either perform mitosis or meiosis. This video will be showing meiosis, which is the division of the cell to create gametes. Meiosis begins with prophase 1. In prophase 1, chromatin condenses to form visible chromosomes. The spindle, fibers, and apparatus begins forming and will finish formation during prometaphase. Synapsis or crossing over occurs, which will cause genetic variation. Chromosomes form homologous pairs, and they are tetrads as they each have 4 chromatids. Centromeres migrate to opposite poles of the cell, and the nuclear envelope will divide in order to prepare for division. The next phase is metaphase 1. In this phase, chromosomes line up in pairs at the metaphase plate at the middle of the cell, and kinetochores attach to the centrosomes. Independent assortment also occurs. The next phase is anaphase 1. In anaphase 1, spindle fibers pull homologous chromosome pairs to opposite ends of the cell. The cell elongates as kinetochores shorten and non-kinetochores lengthen. In telophase and cytokinesis 1, the cleavage furrow divides the cell into 2 daughter cells.
Now, meiosis 2 begins with prophase 2. In prophase 2, chromatin condenses to form visible chromosomes. The spindle apparatus begins to form. Centrosomes migrate to opposite poles of the cell. Next, we have metaphase 2. Homologous pairs start moving towards the metaphase plate of the cell. Kinetochores attach to the centromere.
In anaphase 2, sister chromatids are pulled apart by spindle fibers as the cell elongates. Kinetochores shorten and non-kinetochores lengthen. The last stage of meiosis is telophase and cytokinesis 2. The nuclear envelopes reform around 4 nuclei, and the cleavage furrow splits the cells into 4 haploid daughter cells. In oogenesis, unequal division of the cytoplasm results in the creation of 1 functional egg cell to be used for reproduction and 3 polar bodies that will shortly die off. Finally, in spermatogenesis, equal division of the cytoplasm results in the creation of 4 functional sperm cells to be used for reproduction. Thanks for watching.